Hi everyone, my name is Sahand, and I'm a postdoc in Mo Najjar's lab at USC. And we're primarily a physics group, and that's why we've been studying extracellular electron transport in Shewanella from a biophysical standpoint, and that's what I'm going to talk about today. So most of you are probably familiar with Shewanella oneidensis number 1. This organism was isolated in 1987 by Ken Nielsen, who's also at USC. And it's very popular because it can use a variety of electron donors and electron acceptors, including solid phase minerals such as manganese and iron oxides. So in most respiratory organisms, the respiratory electrons are around the inner membrane. But in Shewanella, because Shewanella needs to respire this solid phase electron acceptor on the outside of the cell, it needs to get these respiratory electrons somehow across the periplasm and outer membrane all the way to the outside of the cell. In Shewanella, there's a pathway that involves a series of multi-heme cytochromes, which are these molecules that you can see here, that have all these heme groups within them that mediates the long-range electron transport to the outside of the cell. And there's a model that is proposed which talks about the porin cytochrome complex at the outer membrane. So this complex includes 3 proteins, basically. There's one at the inner side, on the periplasmic side, MTRA. There's one on the outside, on the outside of the outer membrane, which is MTRC, and there's a porin, there's a beta barrel inside the outer membrane, MTRB. Now, because of the importance of outer membrane cytochromes in this long-range electron transport, we're going to look at the structure of the first outer membrane cytochrome, the first crystal structure of an outer membrane cytochrome in Shewanella. So this is MTRF, and it's a homolog of MTRC, which you saw in that complex before. So this molecule has 10 heme groups within its structure, and it's very interesting that the spacing between the pairs of neighboring hemes in this cytochrome are all less than 1 nanometer, which means that electron tunneling is possible between these hemes. So the idea is that the electrons would enter the cytochrome at the first site, and the electrons would tunnel from heme to heme, and they would exit the cytochrome at the other side. So it would go from one end to the other. And so there are multiple mechanisms of extracellular electron transport that are proposed in Shewanella. The first one is electron transport through direct contact. So the cell, the surface of the cell would directly contact the mineral or the electrode, and it would deposit the outer membrane cytochrome would transfer the electrons to the electron acceptor. The second pathway involves conductive filaments that are called bacterial nanowires. So these are microns long filaments that go from the cell surface to the terminal electron acceptor, and they transfer all these respiratory electrons. And the third pathway involves molecules that are called electron shuttles. So these molecules pick up electrons at the cell surface, and they diffuse back and forth between the cell and the terminal electron acceptor, and they transfer these respiratory electrons. Okay, so there's 3 main questions that we're trying to answer in our group. The first one, which I think is the most important one, is what is the specific respiration rate of Shewanella cells? So to put it in other words is, what is exactly the number of electrons per second that a single cell has to transfer to be able to survive, right? What is the minimum number of electrons that has to be transferred? The second question is, what is the physical basis of extracellular electron transport in Shewanella? How do these cytochromes actually transfer these electrons within their structure? And the third question that we recently tried to answer is, what is the molecular makeup of bacterial nanowires in Shewanella? So in the lab, we have been able to measure the respiration rates of individual Shewanella, live individual Shewanella cells using an optical trapping device and a microelectrode that is about 10 nanometers 10 microns in diameter. So this electrode would act as a little landing pad, and with the optical trapping device, you trap one Shewanella cell and you land it on the surface of this electrode. And you can actually watch that process happen in this video. So the circle is the microelectrode, and that cell that was swimming by was trapped by the optical trapping device, and now it's being slowly placed, it's being lowered and placed on the surface of the electrode. Right there, I think. Yeah. And as soon as that happens, in the current that you're measuring from the electrode, you can see a sudden jump in the current that is in the order of a few tens of femtoamps. So that number actually translates to about 10 to the 5 to 10 to the 6 electrons per second per cell. So this is basically the answer to that first question that I asked earlier. So a single cell needs to transfer 10 to the 5, 10 to the 6 electrons per second to its electronic substrate to be able to survive. Now, we're going to keep coming back to this number because this is basically our reference for all these rates in the different mechanisms. So in our lab, we also do kinetic Monte Carlo simulations of electron transfer in individual cytochromes. So the example here is MTRF. It's the first outer membrane cytochrome of Shewanella with the solved crystal structure. And so with these simulations, we can exactly measure the rates of electron transfer from between these neighboring heme groups within the MTRF structure. And that means that in the end, we can calculate the overall electron transfer through the cytochrome. And experimentally, we've been able to verify those the results of those simulations using scanning tunneling microscopy, and also using a monolayer of MTRF, the decaying cytochrome. So we go in with the sharp tip, with the atomically sharp tip of the scanning tunneling microscope, and we basically measure the conductivity of every single MTRF molecule. And so what comes out of these simulations is a rate or an upper limit for the number of electrons that a single cytochrome can transfer. So that's about, from our simulations, that's about 13,000 electrons per second, or we can say it's in the order of 10,000 electrons per second. Now, from the experimental measurements that the group at the University of East Anglia have done on proteoliposomes, they have shown that the electron transfer rate per MTR-CAB complex is in the order of, again, 10,000 electrons per second. Okay. So each cytochrome transfers, or that's the upper limit for each cytochrome, that it can transfer about 10,000 electrons per second. And also from the experimental measurements, we know that there's about 10 to the 3 to 10 to the 4 cytochromes per cell on the surface of the cell. So now let's assume 10 to the 3 cytochromes per cell, and let's assume about 10,000 electrons per second per cytochrome. So if you multiply those two numbers, you get about 10 to the 7th electrons per second per cell, right? And remember that number that I showed you before, the 10 to the 6 electrons per second per cell, which was the respiration rate, the minimum respiration rate required. So now this looks like this number, 10 to the 7, is 10 times larger than that. So that means that basically on the surface of a cell, you only need about 10% of the cell surface to be in contact with an electrode or with the terminal electron acceptor for the cell to be able to survive or respire fast enough. Okay, so now I'm going to talk about bacterial nanowires. So in Shewanella, bacterial nanowires were discovered in 2006 by Yuri Gorby and his colleagues. And these bacterial nanowires were produced in chemostat cultures that were oxygen limited. And in the past few years, there have been two types of conductivity measurements on bacterial nanowires. So the first type is where you use an atomic force microscope to measure the conductivity across the width of a single nanowire. And that's the result on the left. So the circles show the experimental data. And the second type was a measurement of conductivity across the length of a single nanowire, which is what you have here. So those are the electrodes, and this is the nanowire going between the electrodes. And a few years ago, we actually published a multi-step hopping model that tries to explain the data that is seen in both of these experiments. And based on the multi-step hopping model, which assumes that there's a chain of multi-heme cytochromes along the length of the nanowire, and there's multi-step hopping between the heme groups within these cytochromes, we can actually see a pretty good match with the experimental results in both of these measurements. So that showed that multi-step hopping looks like a promising mechanism in this case. But there was a big issue with all of these previous conductivity measurements, and that was that all of these were not really physiologically relevant because the samples were always chemically fixed and they were always dried in these conductance measurements. And because of the lack of any in vivo work on bacterial nanowires, we still had important questions remaining about these structures. The first one was, what are they really made of? People thought that they're probably pili, but no one had really studied that. The second question was about the structure. Are these structures actually hollow? Are they packed inside? And what is their physiological impact on the cells when they're produced? And the fourth question was about the mechanism of electron transport along the length of these wires. Now, we had the multi-step hopping model as our proposed theory, but we still needed experimental confirmation for that. So, for the first time in our lab, we did in vivo imaging of live growth of nanowires from live Shewanella cells, as you can see in these videos. So here you can see bacterial nanowires growing out of live cells. And in the second video, you can actually see the cell on the left grow a nanowire and connect to the cell on the right. Right there. So in this case, both cells are alive. So we tried to look at the composition of these structures. We used the fluorescent dyes that are specific to protein, the green one on the left, and specific to membrane, the red one on the right. And surprisingly, we saw that the same structures are actually stained by both the protein and the membrane-specific dyes, which showed that there's probably membrane involved here. So to look at that more carefully, we localized GFP to the cytoplasm of the cells in one strain, and to the periplasm of the cells in the second strain. And we did not see localization of the GFP, of the cytoplasmic GFP along the length of the wires, but we did see the periplasmic GFP lining up along the nanowires. So that showed that there's periplasm inside these structures, there's no cytoplasm, and that means that these nanowires are probably outer membrane extensions. To look at the physiological impact of nanowires on cells, we use the fluorescent dye that is an indicator of the respiration rate of the cell. So the green fluorescence that you see in this video is actually that redox dye. And you can see that as the nanowire is produced, the green fluorescence goes up, which means that the cell is actually respiring faster. And you can also see that quantitatively on the right, where time point 0 is when the nanowire was first observed. And so this again shows a correlation between the production of nanowires and an increase in the respiration rate of the cell. And we were not able to see any association between pillin proteins and the nanowires. That's because we used a lot of different mutants that lack the genes for pillin proteins, and such as delta pillay in this case. And we still saw the nanowires being produced and with a similar impact on the respiration rate of the cell. So the green fluorescence again is that redox, the redox dye. So to look at the mechanism of electron transport more carefully, we tried to see if we can find any cytochromes along these these nanowires. And what we did was we used the antibodies that were raised against MTRC and OMCA, which are the two of the main outer membrane cytochromes in Shewanella. And we actually saw that they are localized along the length of the wires. And in the control strain, a strain lacking the genes for MTRC OMCA, we did not see any localization of the antibody. And so again, you can see the MTRC and OMCA. And so based on these results, we proposed a model where bacterial nanowires are extensions of the outer membrane that contain periplasm on the inside. And they have outer membrane cytochromes along the length of the wire, which they are proposed to mediate the long-range electron transport in bacterial nanowires. So we saw something interesting in some cases. We observed an initial production of vesicle chain. So as you can see here, there's 3 vesicles that you'll be able to see, I think. And then that transitions over time into a smoother and longer filament. So it looks like the morphology of the nanowire is also changing over time. This is, again, a live cell. So it looks like the morphology is changing in vivo, and we were able to look at the different phases of this transition using atomic force microscopy, which gives us much better resolution. So you can see here, it's a clear vesicle chain. Here, it's somewhere in between. It's more of a vesicle chain around here and smoother around here. And this one is completely smooth. And the insets show the live fluorescence images. And so so in studying the bacterial nanowires, we were able to answer the questions that we had. So we found that the composition is actually membrane. We found that these are outer membrane extensions, so they're tube-like. We found that there's a correlation between the production of nanowires and an increase in the respiration rate. And we also saw that the cytochromes are being localized along the length of the wires, which is consistent with our previous multi-step hopping model, but it still needs more work. And so right now, we are collaborating with Grant Jensen and his postdoc, Purna Subramanian at Caltech for doing cryo-electron microscopy and transmission electron microscopy to get higher resolution images of these structures. So an example is here. So we have been able to do correlative fluorescence light microscopy and transmission electron microscopy. So you're looking at the same area of the sample. And we have been able to overlap these. So this is an overlap of the 2 previous images. So you can see the cells, so the dark areas are the transmission electron microscopy cells, and the red is from the light fluorescence. And so we can go in and zoom in on these structures. So the advantage here is that using fluorescence microscopy, you can actually stain these nanowires with membrane stain. So you can be sure that these are definitely made of membrane. And then you can go and look at the TEM image, which gives you much better resolution to look at the structure. So again, here it looks like it's more of a vesicle chain. And in the second example, which is this nanowire right here, it looks a lot smoother. So you can see it right here in the fluorescence image. So that means that it's membrane. And here you can see that it's a smooth and longer wire. And we're also doing cryotomography on bacterial nanowires. So in cryotomography, you can actually get a 3D reconstruction of the structure of your sample. So this video is going to show you is going to go through the sections of this 3D reconstructed image. And you can see the bacterial nanowire right around here. So again, you're looking at the different layers of the 3D image. Now the filament you see there is actually a flagellum. There, and I think there was another one right there. So again, this is the nanowire, and this is the flagellum. So the advantage of cryo-electron microscopy is that the resolution is so high that it can easily differentiate between all these types of filaments. So the three main types of filaments that we see in Shewanella are pili, flagella, and nanowires. And the reason I said this is a flagellum is when you go back at the cell where it came from, where its base is, you can actually see a nice basal body in the membrane. And that's the same for the pili. So you can absolutely make sure that what you're looking at is the structure that you want. And so we're trying to use these cryotomography results to possibly look at the organization of outer membrane cytochromes on these nanowires. So again, these are cryo-electron microscopy images. And so we're hoping to be able to see where these the cytochrome complexes are exactly, and what is their spacing, what is their organization along the nanowires. And in combination with that, we're right now extending our kinetic Monte Carlo simulations that I showed you before on individual cytochromes. We're extending that into a network of cytochromes that is decorating the length of the wire. So the yellow dots that you see are occupied redox sites. So that means they have electrons. So that's where the electrons are. And the red ones are empty. So you're basically watching the electrons propagating from one end of the wire to the other. So in these simulations, if you assume a full packing of cytochromes on the wire, so this is the length of the wire, this is a network of the cytochromes, and if you put them in a well-organized system, you can actually achieve electron transfer rates of up to 10 to the 5 electrons per second. Again, let's go back to that number, the respiration rate of the cell that I showed you earlier. That was 10 to the 6 electrons per second, right? And this is 10 to the 5. So it's comparable with that number, and this is from a single nanowire. And in this video, you can actually watch again the propagation of the electrons from one end to the other. Again, the yellow dots are the electrons, and you can watch how they're being transferred along the length of the wire. So we're combining these kinetic Monte Carlo simulations with our cryo-electron microscopy imaging to get a better understanding of how these cytochromes are mediating this long-range electron transport. So again, going back to the questions that we asked in the beginning, we were able to answer the question about the specific respiration rate of this organism. We were able to look at the mechanisms using our simulations and also our experimental measurements, our individual cytochromes. And we've been also able to look at the bacterial nanowires and their composition, and which also leads us to figuring out the mechanism of electron transport in the bacterial nanowires. And with that, I'd like to thank everyone in the LNJAR lab and all our collaborators who contributed to this work, and all the funding agencies. And thank you for your attention.
We're back on time as well. I'm going to ask the first question, so because I get to because I'm the moderator. These outer membrane vesicles, they seem to eventually form. You showed this series of going from kind of blebs into more of a narrow structure, kind of what's shown here too. Mm-hmm. Does that mean, so what's driving that interaction? Membranes have kind of a shape that they like to be in. Are there protein-protein interactions that might be helping get this structure?
You mean, so basically the question is how are they stabilized, right?
Yeah.
So that's one of the things that we're hoping to find an answer to in the cryotomography imaging. And we don't know right now, but it looks like there might be some proteins that are pinching these cytochromes at the neck, or yeah.
All right, Derek, can you be loud? Sure you can.
These are, so that simulation, that's basically the upper limit. So it's fully packed with cytochromes. So in terms of protein interaction, it's basically as many cytochromes as you can are placed on the. Right. Right. No. So we're trying to get insight into what's the rates are and how we need to organize these cytochromes, what the density needs to be, and that's why we're also looking at the cryotomography. So we don't really have an answer yet, but we're just looking into how these are organized, and we're combining basically these two techniques to figure out.
Probably yes. Probably yes. There's some combination of this, I think. But yeah, there's clearly a lot more that we need to do to figure that out. Let's go for Alfred. There's a microphone there.
Two questions. One, your interpretation and actually the model relies on the cellular respiration rate that you experimentally determined to 10 to the 4, 10 to the 5 electrons per second, right?
The model does not rely on that number.
No, sorry, 10 to the 5, 10 to the 6 electrons per second that you measured on the single cell. So
Right.
what do you feel, how accurate is this number in terms of reflecting a true, you said these cells are viable in order to survive. How do you know that this is actually an accurate number as opposed to an artificial number that you measure on a cell that survived the experimental treatment of putting it down in there? How do you know that this rate is sufficient for, let's say, growth?
No, I didn't say this rate is sufficient for growth. It's sufficient for just survival, I think.
How do you measure that? How do you know?
Well, so the current is actually sustained on the electrode for a long time. When the cell contacts the surface of the microelectrode, the current does sustain over time.
Yes, but you said survival. That's different than metabolism.
The answer is I don't really know what's going on in terms of the metabolism, but we know that that's the number of electrons that they do transfer over time. I don't know if it means growth. It probably doesn't because it's a very small number. If it needs
Right. So that's why I'm wondering. I mean, if you're measuring like 1% or 0.1% of the in vivo growth rate, I think it's interesting what you measure, but that has different implications.
Right. So I think we're more interested in the lower limits of how many electrons does the cell need to transfer to just stay as it is and not die, I guess.
Well, you don't know that. That's what I'm trying to say. You don't know whether the cell is actually dead. It could be biologically, it could be dead.
Well, yeah, that's true. No,
Right.
but in terms of if it can sustain this basically current, this number of electron transfers, so that means it's not dying, right? Because over long term, it's...
You're measuring something different. We can discuss this
Okay.
later, but you're measuring something different.
Okay.
The second question, what percentage of the surface area actually relative to a total cell is extruded as a filament?
We have calculated that number. It depends because the thickness of these filaments is variable. Sometimes they're very thin, sometimes they're very thick. I don't remember the exact number, unfortunately, but I know that we have calculated.
I'm wondering where this basically volume or the surface area is stored when there are no filaments.
Can you ask that again? Can you say that again?
Yes, so if you extrude a filament, that
Uh-huh.
requires more surface area to cover that. Where is that in the cell if there's no filament?
They actually do get smaller in a lot of cases.
Okay,
Yeah, we can actually watch them
Yeah.
getting smaller as they produce the nanowires. Yeah.
thanks.
I think in the interest of time, let's thank Sahan again for really. At the break, definitely come up and ask your other questions. Yeah, this is really fun stuff.